So I'm excited to start session 5, which is the climate change and carbon. They're excited too, clearly. And our first speaker for today is Dr. Michelle Tedder from the University of KwaZulu-Natal. And welcome.
Right, I'm going to have to bring that quite a bit lower. There we go. Okay, I'm just going to make sure that I actually can drive this thing before we get started. There we are. Okay. Good morning, everybody. Thank you for getting up and getting going early this morning. I will do my very best not to do anything faux pas worthy during this presentation. Oh dear. I'm going to be presenting the results from our drought trial. Quite a number of you came to see it on Monday, so thank you very much for joining us for that tour. And as we said, we sometimes get visitors on the other side of the fence at Ukalinga. So these are some of our co-researchers. So as we know, grassland biome in South Africa is one of our largest biomes. It takes up 1/3 of South Africa's terrestrial surface. It has the second highest diversity after fynbos. And we expect in terms of climate change that we are going to experience more frequent droughts and heat waves. Our mesic grasslands at the moment, according to current models, are expected to be 10 to 15% drier. We're going to, with this, see biome shifts. We're going to see range contraction, and we are going to see increasing biodiversity loss with warming associated with that climate change, resulting also in decreases in food security, increased economic losses, and overall, we're expecting to see a decrease in the area of the grassland biome through that range contraction of anything from 30 to 50%, which is quite a terrifying thought. If you have a look at this little graph in the bottom here, we currently, at our present state, have 60% of that grassland biome already transformed, and 37% of it is under agriculture. And this tiny, tiny little section of the pie here is what is actually formally conserved. So we are in a pretty worrying state already. So we all know where we are. This was also presented at the International Rangelands Congress, so that was to help give some scale, but our trial is just on the other side of Pietermaritzburg, near the university. And for those who didn't visit, this is what it looks like. We've got 3 blocks because we have a very slight slope on that trial. So we have 3 blocks to encounter or to allow for that gradient. And we have warming chambers set up across that entire trial. We have drought shelters set up to intercept 50% of the ambient rainfall. And you can then see here, obviously, we had to do something with that rainfall. And so to make sure that we didn't end up with an erosion gully next to our trial, we piped that rainfall onto the next plot. And so that obviously becomes a 50% addition plot next to that, which was quite convenient because there are predictions that state that there will be increases in some areas, and some areas are getting wetter. And we've already been seeing that in the last year or two, that certainly around Howick, around areas of Maritzburg, we are having higher rainfall, we are having a longer rainy season, we're having a lot of out-of-season rainfall. So we have those 3 rainfall treatments. We have our warming chambers, which raise the ambient air temperature about 2 degrees. And this trial was established in 2019 and has been running now for, this is our 6th year. So what I'll be presenting today is data from the first 5 years of this trial. So just to give you a little overview of our climatic conditions over those first 5 years, you can see that our rainfall fluctuates quite a lot. So first year we were in the upper 600s, we then increased to the 800s, 900s, then we started to drop again to the upper 700s and then the upper 600s. So we do have a fairly large fluctuation of total rainfall over the year, growing season to growing season. So if we have a look at the species diversity from the first year of that trial in the light green, we can see we were coming out of that hectic 2014-2015 drought where we saw significant effects on our grasslands. And so we set this up a couple of years after that. And so we are seeing a bit of a shift, which is possibly recovery from that drought in 2014-2015 as we progress through the 5 years of this trial. So we have seen a bit of a shift in terms of our species composition. So that is our one key significant difference, is that we have seen an improvement over those 5 years, or change. And we have seen an increase in species richness, as you can see on this graph here. However, that hasn't translated into an increase in diversity. So that's not affecting those key dominant species, which you can also see here on our temporal stability of our biomass. The grasses, as we'd expect, are very stable. Our legumes are our least temporally stable, which makes sense. They are more rare in our grassland. They're generally very small plants, so they don't also contribute a lot to that biomass. And so it's likely that these smaller species and these less abundant species are the ones that we are seeing overall coming back and increasing that diverse or that richness, but not yet affecting the overall diversity. Then if we look at our drought, and we look at the holy p-value of 0.051, and you run an analysis and you get 0.051, and you want to kick something because it's not significant yet, but it's pretty close. We can see a trend. So this is the first time we have been so close to the p-value of 0.05. And so I expect that we are seeing an effect coming through now. So in this, when we analyze the data for the 6th year, I expect that we are probably going to see a difference. And what we are going to see is this drought treatment here is pulling away from our ambient treatment. We can see that that run on, there's not a lot of effect. It's quite scattered. The species composition covers both of these other two treatments, and it's quite varied. But we are seeing this drought treatment pulling away and starting to differ. And so I think in the next 2 or 3 years, we are going to see a clear difference between that ambient and that drought treatment. But as I say, at this point, not just yet, but I'm pretty certain that it's coming. This is where we've been seeing a difference from early on. The warming within the first couple of years started to have an effect on our species composition. You can see it's much, much more varied in our warmed plots than on our ambient plots. You can see that we've had a significant loss of species richness here, but again, not affecting our diversity. So those are our rare species. Looking at the temporal stability that we saw earlier, it's probably those smaller legume species and some smaller forb species. So this is data that going forward, I'm going to dig into more and look at those functional groups and which are the ones that appear to be being affected, because it's obviously not our grass species composition, which is our dominant composition. Now, if we look at the phenology of the grasses, this is first 2 seasons from one of my PhD students. And you can see here, as we would expect, it's very, very strongly linked. Your onset of budding is very strongly linked to the onset of rainfall, as we would expect. But what we did find is here, this is your onset of budding, which was delayed in your drought treatment. So it's not just the presence of the moisture, but it is the amount of the moisture that appears to be important when it comes to your onset of budding. And so drought delayed that onset of budding by a couple of weeks. And then obviously it makes sense that that onset of the flowering then was a knock-on effect, and it was delayed as a result of that. So as we start to see more frequent drought, we're likely to see later budding, later greening up of our grasses. Even though we're not seeing a reduction in abundance of them, we are going to start seeing shifts in that greening up, and as a result, the seeding and general phenological processes. Then if we move on to the forbs, just for you, Cindy. So this is work from Tandwayo, who presented yesterday her PhD proposal. This is work from her master's that she did on that drought trial. And we can see what to me are some quite terrifying results. So these are 2 of the species that she focused on. And we can see we can see under the drought, if you look at when the species were flowering, there's certainly a shift in Thunbergia where it pushes much later. But this is what really worries me. We've got 3 flowers over the growing season on the plants that she was observing in total count, compared to the ones in the run on where we were seeing a total count of 15 on the ones that she was monitoring. And then if we look at Cephalaria pungens, we're seeing the same thing. We're not seeing as much of a shift in terms of timing, but we're seeing the same reduction where we had 37 inflorescences total in what she was monitoring, compared to 136 and 163 in the ambient and the run-on plots. So this to me, in terms of our pollinators, in terms of our grassland forbs and their recruitment, I find very, very concerning. And it's something that I think we need to look into in a lot more detail to see how are our forbs that contribute the most to our grassland diversity being affected by climate change. So overall, what did we see? Rainfall manipulation at this point, there hasn't been a significant effect, but it's definitely on its way there. It's coming in the next couple of years. So my estimate, we're looking for these mesic grasslands at a resilience of 5 to 6 years, maybe 7 years, before you start to see those significant changes in your species composition. There hasn't been an effect on richness in terms of the drought just yet. We haven't had an effect of diversity on the drought, but we were seeing that in terms of the warming. There is this delay in budding and flowering, which obviously, if that exacerbates, that becomes more of an issue. It is a concern for your livestock production. It is a concern for stability of those resources. And we're seeing this decrease in forb flowers, as I showed you, which is very concerning in terms of our pollinators, in terms of our general forb diversity and survival in those systems. And the warming, as we saw, we're seeing increased compositional variability. I want to dig into that data more and see what exactly is it that's coming in. Are we seeing exotic species coming in? Is it more weedy species that are coming in and causing that variability and replacing the species that we are losing? And we're seeing a decreased abundance of a lot of the common species. We're seeing a decrease in richness. But as I say, that diversity hasn't been affected yet. What we are doing now is that we've just taken those warming chambers off, and we won't be putting them back on. We are going to start monitoring the recovery on that and see if we start to move back towards that original species composition, and if we do, how long does it take? So what's our take home from this? We all know vegetation degradation is going to likely with extended drought exposure, and we can see that this is likely to be exacerbated by warming effects. So where do we go now? I am currently working on establishing a drought network within South Africa. So, sorry, this project I didn't mention in the beginning is part of the International Drought Experiment. And so there's over 100 sites all around the world that are using the same experimental design, but there are not very many in South Africa. There's only one other one that I'm aware of as part of the drought network. And so we're wanting to try and increase that. And so we've already set up a site in Clarence, just outside Clarence, working with University of the Free State, and sorry, there it is, just outside Clarence, working with University of the Free State and working with Centre for Biological Control, where we're also going to be working on some alien invasive responses to drought and warming. And then we are busy talking to staff from Solplaatje University and also in collaboration with University of the Free State to set up a site in Kimberley. And we're really keen to talk to anyone who is interested in hosting a site to increase this network so that we can get a range of sites across our grassland and savanna biome so that we can look at some broad scale effects on as a result of drought and warming in South Africa. Thank you.
There's time for a question. Would anybody like to? Yes, is there?
Nice present.
There we go.
Great. Thanks for the presentation. Is this thing on? Okay. So the idea of drought, of course, we are all worried about it. But
sorry, I'm struggling to hear you.
I say, you know, the continuous drought is something we all worry about, but under climate change scenarios, the evidence seems to suggest more variability. And so episodic events are not just drought, but also very high rainfall. Are you incorporating that in any of the research? Do you see what happens if you get periods of dryness and then periods of extreme wetness?
Not as yet, but that's something that I am interested in expanding towards.
An online question from Craig. Hi, Michelle. What is predicted to replace the declining grassland biome?
So they're expecting a lot more of the drier sort of biomes, and most of the predictions are linking it to similar to the Karoo type of vegetation from what I've seen so far. Okay, thank you.
Our next speaker is Dr. Heidi Hawkins from Conservation South Africa and the University of Cape Town. Welcome, Heidi.
Thank you, Robin. Good morning, everyone. So I've added a bit to my title, Not Seeing the Grass for the Trees. We're going to talk about grass-associated increases in soil organic carbon via certain fractions. So we all know afforestation is a climate solution, right? More than 60% of African countries, which you see there in red, have committed to restoring millions of hectares by 2030 in deforested and degraded lands. And there's a lot of money behind this, so it is happening. But I think most of us here understand that there's a problem. And that is that there's a misclassification of grassy biomes. And you see tropical ones there in orange on the right. I can use my pointer. Oh, no, I have to point there, yeah. And then if I overlay that AFR 100 map, you see that the target areas are primarily ancient grasslands and savannas that have been misclassified. Those are the striped areas. So this tree planting, but of course woody plant encroachment, we all know affects biodiversity. But the leaders of these countries don't even have the evidence that the planting is going to result in more carbon where it really counts in our biggest terrestrial carbon pool in the soil. So this study is looking at how do grass tree shifts affect soil organic carbon and its stability with rising CO2. So myself and the second author, Mike Kramer, we were lucky enough to join an existing experiment at the Rhodes Elevated CO2 facility that you see there on the right with the open top chambers. And it was a pot culture in a local sandy cambisol. And then we had 400 ppm as ambient and then 550 as elevated. And the treatments were either tree, and we used one of 5 leguminous Vachellia or Senegalia species, or the C4 grass, the Nida, or both of them grown together. And then we were testing 2 ideas. The first hypothesis was, well, we think when you've got trees and grass grown together, that this will really favor fast decomposition because you've got a lot of biomass from the grasses and nitrogen coming from the nitrogen fixing trees. The second hypothesis was just looking again at the idea that CO2 fertilization at high CO2 is going to favor either the C3 trees or under some conditions, if there's water limiting, it could be the C4 grasses. So what do we find? On the left there, we've got soil organic carbon in grams of carbon per kilogram soil, so the concentration. And then on the X, we've got trees alone, grass alone, or grass plus trees. And what you can see is that when we've got grasses, not trees, that you get an increase in soil organic carbon over the 2 years. So that is quite an impressive increase given that soil carbon takes quite a long time to change. And the same thing for soil organic nitrogen. And then surprisingly, when we had the 2 growth forms together, we didn't see that increase. And then for the second hypothesis, CO2 had no effect and no interactions. And you'll see that for most of our plots. So what's going on here? We had a look at the biomass, and you can see just generally that the grass biomass, whether grown alone or in competition, was higher than the above ground tree biomass. And it's even more obvious if you look at the photosynthetically active, just the leaf biomass. So we're beginning to think, well, the grass-associated increase in SOC that we saw is related to biomass rather than some specific aspect of the grasses. Again, no CO2 effect. So then I mentioned the stability of SOC. First, we're going to have to say a few words about soil organic carbon fractions. So if you start up here with dissolved organic matter, which comprises organic nitrogen and carbon mostly, for example, in root exudates, then those are very quickly utilized by soil microbes and then get fixed onto clay mineral surfaces, which you see there. And because there's limited surfaces there, that carbon can saturate quite quickly, but it is very stable because it's bound. And so it has a very slow cycling and long lifetime. On the other hand, you've got particulate organic matter, which is literally pieces of decaying matter in the soil, and they're not bound to anything. They can accumulate indefinitely, but because they're not bound, they can also be lost quite quickly. And then a lighter part of that POM is the light fraction. So what did we find? Well, nothing when it came to dissolved organic matter and mineral associated organic matter. Also nothing with the CO2 and nothing with the light fraction. But if you look over here, you see a similar pattern for the POC, the particulate, as we saw for the bulk soil. So it looks like the grass-associated SOC increase is due to that particulate organic matter. And I'm just showing the carbon, but it's the same for the nitrogen. No effect for the CO2. So what we did then was we used an end-member mixing model where we're trying to get at, okay, how much of this soil organic carbon is coming from the grasses. And just briefly, here you use the stable isotopes of carbon and look at the difference in their values between C3 plants and C4 and in the soil. So I won't go into details there, but what we found was that if you look at 400 ppm, you can see that the C4 grass, the amount coming from grass, is really high, around 80 to 90%, whether you're looking at SOC or POC or MEOC. And it appears to even increase under elevated CO2. This is for the light fraction. It's also a lot coming from grasses, about 50 to 60%, but then obviously there's more coming from trees there. So exactly what these percentages are and the CO2 effect for us is in question when we're using these end-member mixing models, also for other studies, because of technical issues. So if anyone's interested in this, you can chat afterwards. But the point for us is that no matter what that exact percentage is, the C4 grasses are probably contributing a lot to all the fractions. So then we went and did a structural equation model, and then the role of the POC in the SOC change was also strongly supported by that. So I'll just talk you through that. We've got it's a very simple one. We've got above ground biomass, below ground biomass, and then the soil organic carbon, and then our POC and our MEOC. And I just want to point out that in the mineral associated fraction, that is where most of the carbon is sitting, and that's very common across the world. So these savanna soils were similar in that respect. And then we've got the blue arrows showing the pathway of the processes, and next to the arrows, we've got the regression coefficients. And so just very briefly, what we see is that especially the POC is playing a role in the SOC formation with its high coefficient there, and it's largely via the grass biomass, especially above ground biomass, which surprised us. And then, you know, in this whole talk, we haven't seen a response from the MEOC, but it may well be that you could see a response in other soils with higher clay contents, or if we ran the experiment for a longer time. But it could also be that the POC is indeed the dominant pathway in these dry savanna soils because, well, in European grasslands, where a lot of the information comes from, this is the dominant pathway, but there's more rainfall, possibly the grasses are producing more exudates, and so this whole pathway can run. So there's that knowledge gap, and it would be nice to have more data on savanna soils across soil type gradients and rainfalls. And then I wanted to show you this recent beautiful map, of this is the MEOC and this is the POC. And this is a big improvement on previous maps. But when you dig into the data, you find that that the savanna biomes represented by 22 samples. And in Africa, that's only 10. So that's a lot less than the 3,000 odd for certain forest types. So it's not really adequate to do a map like that. So what's the significance of what we've seen? Grass-associated SOC increases via the particulate fraction. We need more savanna SOC data for earth system models. In terms of soil conservation, we really should take care to avoid erosion and plowing. We don't want to do that anyway in savannas, but also to manage natural disturbance, because this POC fraction is responsive to land use change, but it can also be lost. And then for the same reasons, for carbon offsetting, if you do have a project, then models need to include these fractions to capitalize on nuances, because in the beginning of a project, you might only expect to accumulate this vulnerable fraction. You should know that you're going to have to wait decades if you want to change that stable fraction. So we've got more to learn, but we don't have to wait. Meanwhile, we have clear win-win, many of us do, for policymakers, and that is conservation of grassland biomes, not tree planting, aligns with both biodiversity and climate goals. So I want to thank the roads team for allowing us to do the soil sampling, also the funders, and to you for listening. Thanks.
Time for our questions soon.
I'm going to do a big ask here. I was involved in the UNCCD meeting in India a few years back. One of the problems is there's a misclassification of vegetation types in their system. So they classify savannas as forest, and I tried to argue it. And we need a very strong argument against that to actually change it. The second is with the new, with next year being sort of a rangelands year, can we not put together as a society a very strong policy brief, firstly to DFFE, and then force it through the systems to UNCCD, because they're advocating two things: a strong land tenure system, which we heard yesterday is not necessarily a good idea, and they're not listening to this. We had an intervention where we were saying it's stupid to put your things above ground because of fire. You didn't touch on fire, and I think that might have an additional impact there, and it's certainly matching what we're seeing at Cathedral Peak, your results. So I think we need a very strong intervention, and I think all those working on this should sit together, and an outcome of this conference and the one from last year as well should be a statement going via DFFE, convince them, and then up to UNCCD.
Yeah, I agree completely. And perhaps we should just also check what the Future Ecosystems for Africa group are doing, because they're focusing on policy briefs. But I agree, though, I mean, that we can use that Rangelands International Year platform.
Our next speaker is Reneilwe Maake, and she's from the Agricultural Research Council in Pretoria. Thank you. Welcome. There you go.
Good morning, everyone. As I've already been introduced, my name is Reneilwe Maake from the Agricultural Research Council. I'm also a PhD student with the University of KZN, and I would like to thank you for giving me this opportunity to share with you one of my research objectives, which is focusing on using remote sensing to quantify above ground carbon stocks in savanna ecosystems. And I will contribute to our previous speaker because they were looking at assessing from ground level, and I'm mainly focusing on the quantification and monitoring aspects of it. Okay, so why do we need to specifically focus our interest or research interest into these savanna ecosystems? It has already been alluded that they cover large geographical areas. They are characterized by rich biodiversity, where there is a lot of grasses which coexist with the woody layer. And both of these layers contribute significantly to the accumulation and sequestration of carbon dioxide from the atmosphere. And now we have also learned that disturbances such as fires and the removal of grass for harvesting are contributing to the loss of this carbon dioxide, which also contributes to the increase in climate change. So we've also learned that seasonal dynamics also have an impact on this accumulation of carbon in these above ground carbon stocks. So we need now to observe these dynamics over seasons as well. So in the past, or rather currently, some of the researchers are still using ground-based measurements, but we have learned that these are limited in terms of the area that is covered. They are also expensive to implement, and they require a lot of labor. And also the long-term monitoring efforts from those ground-based observations are fragmented, and they are inconsistent in some extent. So remote sensing offers us another different opportunity because it covers the large geographical areas, and we can also frequently observe these changes over time. And we have also learned that it's cost effective, and we can also access areas that were not inaccessible using ground-based. So why do we now need to quantify these above ground carbon stocks? To inform greenhouse inventories, to also provide insights for international agreements, and also quantifying, or rather understanding the distribution of these above ground carbon stocks also helps us implement the Sustainable Development Goals, which is 13, which calls for immediate combat in climate change. So now zooming into the remote sensing, what are the challenges that we face when we estimate these above ground carbon stocks using these sensors? We know that we have optical sensors, we also have SAR sensors, but optical sensors are limited by cloud. Today it's cloudy, so we cannot capture the images because the sensors cannot penetrate through the clouds. And at night, we also are limited in capturing these images because it mainly relies on the light. And also, it's also insensitive to dry seasons when we look at seasons. So now, which means we are limited when we do monitoring studies because we cannot focus on the dry season because it depends on the chlorophyll or the greenness. So SAR data as well is also limited because it looks at the structural characteristics of the grasses, and also it requires expert knowledge to be able to interpret those images, and it's also sensitive to soil and wind. So now what do they share in common? So we have also learned that they also saturate, especially when we observe or monitor these carbon stocks over dense savanna ecosystems, and also changes in seasons, high moisture content, vegetation structure also affects this. So now we are forced now to look at alternative ways now, which calls us to this study where we are looking at determining whether integrating these variables that we extract from SAR, as well as texture and topographic variables can improve the prediction of this. So the study was conducted in the Kruger National Park, which is characterized by the woody as well as the grass layer. And we conducted field work in February, which covered the wet season, and July, which covered the dry season. And during those two periods, we demarcated plots. We cut the grass, we weighed the dry weight, we dried the wet samples, and then we weighed the dry weight. And on the remote sensing perspective, we acquired the SAR images. We pre-processed for the 2 seasons, we pre-processed them. We extracted the bands, we calculated the indices from the bands, and then we extracted the predictor variables, and then we now trained the models using those variables. So these are the predictor variables that we looked at. We looked at them as texture metrics. We looked at the indices extracted from the SAR. We also looked at the topographic variables. So we opted for the cutting edge machine learning algorithms, which is one of the machine learning algorithm, which is the extreme gradient boosting. And then we regressed the samples that we collected, and then against the variables that we use to predict the grass carbon stocks. So we set up 6 experimental configurations where we looked at each of those variables individually and assessed the performance, and we also looked at how they would perform if we were to combine. Does the accuracy increase? Does it decrease? And whatnot. We also, because to ensure that our model are performing well, we have to now compute evaluation metrics. We looked at the coefficient of determination. We looked at the root mean squared error. We looked at the mean absolute error. Now, don't be frightened by the table. I'll quickly jump into, okay, I cannot see there. But what the key findings is that when we looked at those individual predictor variables as standalone variables, when we fed them into the model as standalone variables, they tended to produce weaker correlations, and as opposed to when now we combine, because now it means that they bring in complementary traits or characteristics where if the other variable is failing to pick up some differences within the carbon stocks, then the other one is able to complement what is missed by the other one. We also looked at which variables amongst those ones that we used contributed most to the model. And we found that the radar vegetation indices consistently performed or contributed to the model, and then the topographic variables such as the TSIR also contributed. So now what does this mean? It means that on standalone, because we know that the world is a system and it's interrelated, so we need this interrelated approaches where you combine these factors that complement each other so that you can get better results. And we've also learned that seasons also contribute to the performance of these models. And we've seen the dry season showing superior predictive power, sorry. And then moving to the future, I will look at how this model, because we also need to know how, if we can transfer this model to other temporal resolutions or across different years so that we can see how they perform. Thank you.
Are there any questions?
Thank you. We all know that these days weather is really unpredictable and very extreme. why did you choose weather as one of your variable? And apart from the weather, what other variables maybe did you use? I saw there were a number of them, but I was just concerned about weather since it's very unpredictable. Thank you.
I didn't use weather. I'm not sure if I got the question correctly. Did you say weather? I didn't use, sorry. Oh yes, we just wanted to see how the model is performing across the two seasons, because obviously they bring in different environmental conditions. So we wanted to see how the model performs in the dry season, which is influenced by maybe rainfall, temperature decrease and increase, and also in the dry season as well, so that we can be able to see if we can be able to transfer the models across those different conditions, because that's basically what we need in remote sensing. The models are technically intransferable. It's either they are site specific or they are dependent on certain conditions for different environments.
Time for one more question. I saw a hand at the back earlier. Oh,
I've got the mic. So. Hi, thank you for a very interesting presentation. I was wondering if you've thought about using something like adjusted R-squared or best subset selection using Akaiki to compare the models, because I think the way I understand it, the model with the most predictors was the best model. Am I correct?
The what?
The model with the most predictors was your best model.
Yes.
Yes, but whenever you add more predictive variables to your model, you will always get higher R-squared. So if you use something like an adjusted R-squared or an Akaiki, then it penalizes for those extra predictors, but it gives you maybe a better idea whether it's just a statistical effect or whether it's an actual effect. So I was just wondering if you had considered that.
I will consider it with your help going forward. Because I'm not from a statistical background, but yes, I've also noted that those matrices also contribute. They give better interpretation of the model. But if you are willing to help, please. Thank you.
Our next speaker is Professor Tony Palmer from the Rhodes University. Welcome, Tony.
How are we doing time? Are we just about spot on, eh? Hi. Let's see if we can get this thing the right way around first. Hi, good morning, everyone. It's lovely to be here. And thanks to the organizers and everybody for making it possible for us to all get together again. It's great for me as a retiree to eventually be back in the fold, as it were. But what I'm going to do today is I'm going to talk about work that's been conducted on our research site in the Eastern Cape. And there's quite a large team have been involved here. It's not just Craig and myself and Kathleen, but there are a number of people here in the audience, Buko and Andizwa and Tandazwa, and many others who've been involved in collecting the data at the site and establishing it. But I will explain more about that. But thanks to them for the help that they've given in establishing a baseline that we have for this work. All of you know that woody encroachment is a huge problem throughout the world, and these big blue dots on this map show you the extent and the nature of the problem around the world. And as you can see, it's particularly marked in southern Africa. And we've all been grappling with this problem for many, many years. And I hope today we managed to give you a slightly nuanced perspective on what some of the issues that we need to consider when thinking about woody encroachment and the reforestation that is being planned worldwide. Our key questions that we're asking are, what are the differences in mass, which is essentially the carbon, and energy, which is the water fluxes, over two structurally different Southern African savanna how do we, and grasslands, how do these differences help us to better understand the impact of woody plants on native grasslands? And is drought a possible explanation for the heterotrophic respiration that we have experienced at our site? So there's an example from Andrew Scano's work of the extent of encroachment in Southern Africa. And you can see the south on the diagram on the right is the one that's giving you the worst encroachment by Variability karoo there in the southeastern Cape and in the southern escarpment grasslands. Okay, so our study design was to establish two identical open path eddy covariance systems about approximately a kilometer apart from each other, one being located on a site that was invaded by a kosher karoo or Variability karoo, if we want to be American or Australian. But we were able to establish these two permanent towers for a period of nearly 2 years, 651 days, actually. And these towers, for those of you who are not familiar with eddy covariance, provide very high resolution measurements of carbon, of CO2 flux, and the evaporative flux above the canopy. And this exchange enables you to reduce these down to 30-minute intervals and to integrate them over days and over several hectares. So here's an example of or picture of the site, and you can see the encroachment. The VKA is the Variability karoo site, and the GRA is the grassland site. And you can see that the tower on the right is the one at the Variability karoo site, and that is about 2.8 meters above the top of the canopy. So we had quite a lot of fun, Kathleen and Craig and myself, climbing up and down the tower with safety harnesses. And you can imagine there was lots of anxiety from home about my doing this, but it was a very interesting thing. This is the history of the site. So you can see that on this particular farm called Endwell, there's the early photograph on the top left-hand corner shows the site in 1947, I think it is, 49. And then the encroachment happened, but you can see it didn't happen in that patch of grassland at the end of the, on the bottom right-hand corner. Is that how many minutes I've got left? Is that right? Is that correct? Goodness. Okay. Okay. Well, flip up that side. There's the history of the climate at the site. There's the rainfall and the recent, the mean annual rainfall on the left and the long-term figure on the right. You will see that there's a drought period mentioned here, and this is the period of our flux measurements. The data analysis, we analyzed according to this massive amounts of data. We analyzed with the ready proc, and we were able to obviously establish the standards that had been, or use the standards that had been recommended by the community. Our results, as expected, both systems generally functioned relatively similarly, and this was not surprising in terms of carbon and water regulation. The near complete failure of one of the sites in the late December of 2019 corresponded to the end of a multi-year drought, and we had some data losses there, but they weren't too significant. Here's the data from the biomet data showing how temperature varied at the sites and relative humidity and vapor pressure deficit. I need to point to the right thing there. Okay, the energy fluxes, essentially the growing season evaporative demand was calculated at a ratio of ET to precipitation, and they were almost identical. with effectively all of the rainfall being utilized in the dry seasons. The water use efficiency, which is GPP over ET, were essentially identical at both sites, with the site accumulating 2.7 grams of carbon per meter squared per millimeter of water. At the GRA site, it was marginally more efficient in the dry season. However, the growing seasons at both sites were identical, 2.8 grams of carbon. This is a much more complicated diagram to unpack, but you can see the NEE, that's the net ecosystem exchange at the bottom. That's the grassland site, the grassland site and the Verchelia site. And it's clear that the grassland site accumulated more carbon. So that means that means it's a sink, and we're seeing a carbon accumulated into the system at that level. The top line is the GPP, and that's the NPP. So we're seeing quite a small difference between GPP and NPP at the grassland site. And this generally shows us that we are experiencing more respiration at the Verchelia Karoo site. I won't, I've only got a short time left now, but there's a lot of data to unpack here about GPP and NPP and respiration at the ecosystem level. And when Craig has been analyzing this data, he's picked up that we're actually seeing a greater net R-eco at the Verchelia Karoo site. And we're attributing this to an increase in or a high level of heterotrophic respiration at that site. And heterotrophic respiration in these systems is essentially driven by those organisms that we find in the soil, an enormous number of earthworms, termites, and of course the things like mole rats that turn the system over all the time. And Heidi has been mentioned the role that disturbance plays in chewing up the carbon in these systems. And I think that we have really neglected to understand what the role is in heterotrophic respiration in the sites that have a big supply of carbon. And as the Verchelia Karoo dies off in droughts, as happened in our case, there's suddenly an abundance of carbon in the surface, and Craig's analysis shows that we are seeing an enormous heterotrophic respiration signal relative to the grassland, which, if we went back to the biomet data, I could show you, is much warmer and much drier than the understory of the Verchelia Karoo invaded site. So we have this combined effect of basically a cooler, moister site against a hot, dry grassland site. So there's a better explanation of this, where we managed to calculate the heterotrophic respiration, and we are confident that that's the reason why we're seeing this reduction in the carbon stored by the savanna as opposed to that stored by the grassland. And these are examples of some of the drivers that we expect to be able to unpack a little bit more. But essentially, in summary, drought is sustaining pulses of heterotrophic respiration, which can last for years. This drought exposes carbon, and there are risks of large episodic loss. Globally, woody encroachment is thought to be a sink in most cases, but in episodic systems like this one, it moves to be a source, and the vertical shift to carbon above the ground makes it more vulnerable to loss by heterotrophic respiration. And I'd just like to conclude by saying that despite high levels of productivity linked to these increases in wood biomass, the grasslands are significantly sequestering more carbon than in the woodland. And if anybody wants to unpack this some more, we can do this. I see my time is up, but I'd just like to say thank you to the team and to the National Equipment Program, the ALC, Roads, and the NRF for funding this work.
We've got time for one question. Is there questions from the floor? Yes?
Tony.
Thanks, Tony. You didn't mention much about the grass biomass at the encroach site. I was just wondering if the sort of increase in woody biomass is matched by decrease in grass biomass at the Victoria site.
We have all that data. I wouldn't be able to show you or tell you offhand whether there was very little grass herbivory taking place, and we've measured all of the grass biomass that has accumulated. I can't give you an answer without having a look at the data. I'm sorry. We could look at it together afterwards.
Our next speaker for today is Dr. Michelle North from the University of KwaZulu-Natal. Welcome.
Good morning, everyone. Oh my word, that time is scary. Anyway, so today I'm going to be just presenting some of the results of a systematic review that I've been doing for the past few years. It's not yet published, but it's getting there, where we are looking at how African livestock keepers or farmers are responding to climate variability and change. Just before I start, I want to acknowledge my colleagues, Dr. Nina Hunter, Kevin Queenan, and Rob Slotto, who have helped get me through this process, as well as the German Academic Exchange Service and the Wellcome Trust, who are responsible for the funding. So just to start with our question, how African livestock keepers responding to climate variability and change. As we know, climate change is already impacting, well, most of the world, all of the world, but Africa is experiencing altered seasonal precipitation, increasing consecutive dry days, increasing average and extreme temperatures, and many other things. And in Africa, because there's a large dependence on rain-fed agriculture and rain-fed grazing, and also a lot of social and economic factors have contributed to people being highly vulnerable to climate variability and change. And I know these maps at the bottom from the IPCC are really small. The point is not in the details, but I think the main take-home message from these different maps looking at extreme temperatures, droughts, and soil moisture is the fact that the climate is changing and is projected to change variably across the continent. So we're not looking at any single changes across the continent, but some places are getting drier, some places wetter, and therefore the impacts on livestock will be different in different areas, and people will have to respond differently. And then when we look at the animal component of the research question, as has been admirably introduced yesterday and this morning, livestock are critically important for food and nutrition security, livelihoods, resilience, country level GDP. And as we also know, livestock are highly vulnerable to many different climate hazards. And what we noticed during the last IPCC assessment is that in general, but also Africa specifically, there's a huge gap in the literature when it comes to adaptation and impacts of climate change on livestock when compared to crops. There's an abundance of literature on crops, and livestock are largely understudied. So to start approaching this research question, we applied a systematic review. Because African literature is often not incorporated into some of the major databases or into the major global reviews, we tried to cast the net really wide. So our search string was very comprehensive. We had a lot of search terms. We translated them into French as well as English, all different synonyms. And then we also made sure that we included a wide range of different databases to try and include as many local journals as possible as well. Sorry. Then when we screened the articles sequentially by title, abstract, and full text, and applied predefined inclusion criteria. From the included studies, we recorded data about the publications themselves, about the studies, and then also about all the responses that farmers were implementing that were reported in these different papers. And then in terms of the responses, we coded and classified according to a range of different criteria. So just when we look at the results in terms of the actual articles that were included, there were 186 articles that were included that were published between 2014 and 2022. As you can see on the right, there was an increase in publications over time. However, the growth was not as rapid as we've seen in a lot of other climate change related disciplines. Unfortunately, the graph at the bottom is not other disciplines, it's my initial search results, but you can see that there is an increase, but it's quite steady. So I apologize for the slightly busy slide. If we just start on the top left, where we look at the focus of the different studies, we can see that most of the research has taken place in East Africa, particularly Ethiopia and Kenya, followed a little way behind by South Africa, which is strange. I would have thought South Africa would have had more. What's very interesting about this body of research is that the authors, the lead authors, are predominantly from Africa. 80% of the lead authors are from Africa. And this is really different to a lot of other big, especially climate change related disciplines where you find that most of the work is parachute science, where Europeans or North Americans are coming into Africa and studying African systems. Here it seemed like there was a lot of local interest and expertise. And when you look at the red map, it was also interesting that a lot of authors seem to be studying sites in their own countries, particularly in South Africa, Ethiopia, and Kenya. Underneath my selfie, I've just noted that there's a massive gap in research over Central and Northern Africa. And I know this is true for many different disciplines, but I think this is a very big thing to highlight, that we need a lot more research from these countries that are currently absent in the literature. When we get to the responses themselves, obviously, because there's most studies from East Africa, we can see that most of the responses that were reported were from East Africa. But this will also then bias the types of responses because of the types of systems that occur there. And then if we look at the climate drivers that we see across the board, other than West Africa, the most reported driver of farmers' responses was drought. I mean, I know this is not surprising, but what was interesting is Western Africa, they really highlighted climate variability was more important. And then you see that there's some other more region-specific drivers, such as increasing temperatures in Northern Africa, although that was on a very small sample. And what was really interesting for me is we've done a similar systematic review on impacts and adaptation in cities. And in cities, urban residents are responding mostly to extreme weather events like flooding and heat, whereas farming systems, I know it's not surprising, but I found it quite interesting that farmers are responding more to these slower onset hazards. And then what I thought was quite fascinating is most farmers are actually not responding to climate change itself. They're actually responding to the more secondary impacts of climate change. So their primary motivation for responding is a lack of crops or lack of feed or forage or reduced grazing. When it comes to the types of responses that farmers are implementing, by far and away the most common were responses that are relating to the management of the herds. So for example, mobility, destocking, or changing the composition of the herds. These were followed by feed or pasture management related responses like supplementary feeding, supplying animals with different sources of forage or conserving forage. And these were followed sort of lagging a little way by what I termed as system change responses. So for example, diversification of either on-farm or off-farm livelihoods. And now the next thing, this is something that actually contrasts quite starkly with what was presented yesterday, was in this body of literature, there was an extreme overrepresentation or overabundance of small-scale subsistence farmers, and very few responses and papers that looked at how commercial farmers are responding to climate change. And I think, so this is something that I keep seeing coming up, and my explanation for this discrepancy between my study and, for example, the work that was presented yesterday is I think it's to do with terminology. I think when you look at the words respond or adapt or coping with climate change and climate impacts, the type of work tends to favor the smallholder subsistence pastoral systems. I think that when you look at manage, when you look at production, those sort of more conscious, economic driven actions, I think if you're using those specific search terms, you're going to find more commercial farmers in the sample. So I think it's going to be quite interesting to see why, because the issue with this, I know it seems very minor, but the issue is when you look at the IPCC reports, the authors are looking specifically for adaptation. And so they are not picking up commercial farmer related literature. Finally, what we also looked at was the barriers and enabling conditions that were mentioned by some of the studies. And I know it's not surprising, but the most frequently mentioned barrier to adaptation or responding was money, whether it was the cost of implementing the response or the fact that the farmer didn't have enough money or wasn't able to access credit. The second most common barrier was a lack of knowledge, either a lack of experience with the different species. So for example, a lot of people were turning to camels, but they didn't have a traditional knowledge on how to handle camels or how to treat them. They also didn't have enough knowledge about what was available, what they could do to respond, or they didn't have any early warning information about the climate hazard that was coming up. The third most frequently mentioned response barrier was in terms of land, either lack of access to land, competition with crops, increasing competition with crops and other land uses, and then also access to migration routes, as was mentioned yesterday. And then finally, there was a lot of mention of a lack of government support, where policies and money were going more into cropping and into fencing off land and increasing crop-based agriculture rather than supporting people who have livestock. So this particular aspect of my systematic review is more highlighting gaps for future research. We are looking more at policy recommendations elsewhere, but I'm not presenting that so much today. So firstly, there's a massive gap from Central and North African countries. In this particular pool of literature, I think there's a massive gap on what commercial farmers are actually doing to respond to, for example, heat or drought. The body of literature was, the quality was, yeah. And so a lot of them were really small scale, small focus, not necessarily very well done surveys of people. So what we really would like is some more deeper insights into the longer term outcomes of different responses, a better assessment of the feasibility and effectiveness of these responses, and then also much more systems approach to to adaptation where livestock are considered as part of a complex mixed system and environment. And yeah, just my time is up. So just to conclude, livestock farmers have very good awareness of climate hazards and are already adapting their systems. We know this. However, what they're doing currently is going to be increasingly inadequate. And so just a call for research that focuses on informing action that's also driven by what farmers are needing or livestock keepers are needing, and then also addressing the geographic gaps. Thanks.
Are there any questions from the floor? Yes, Susie.
Thank you, really interesting. I don't know if you're referring to my talk, but I've noticed exactly what you're saying. There are lots of small studies on adaptations and stuff. I've just been to a drylands conference in Nigeria and to the International Rangelands Congress, and it's what everybody's doing. But what we are lacking is like detailed, in-depth data on the effects of what actually happens, because it's very easy to do a survey and saying, what is your opinion? What is your experience? And I mean, obviously, we do need to build that picture, but what is really hard is actually finding out what is the effect. And those data, I stand by it, for the communal farming sector are like extremely scarce, and certainly in South Africa are not being collected.
Yeah, I would totally agree. And I think my other suspicion is that there's a lot of money for people to go and study and pastoralists, because America loves to give money for that kind of thing. So a lot of researchers, and I'm not going to mention countries, but they go out and they survey and they repeat the same survey here, but none of it collects the kind of data that you're saying, like actually tracking what works, what doesn't work, how well does it work, how long does it work for, does it work if you have repeated climate hazards? Because it's all very well if you respond to drought today, but if you have another drought next year or in 3 years' time, are you able to do the same thing? And we're needing much better quality research coming out. Otherwise, we're not actually informing policy or practice at all.
Great, thanks for that, Michelle. My question is, given the knowledge gap, what is your review leading to?
Sorry?
What is your review leading to?
For me personally, or for the field as a whole?
No, in terms of your research.
For research, well, for me personally, I think it's pushing to take like these list of recommendations. So many organizations have so many recommendations, but actually go to farmers with the lists and say what are feasible, and then also try and do some studies on how effective they are. In terms of the gaps in areas, I would really push people to start designing studies in Central Africa, but funders also need to start pushing to fund studies that are looking. North Africa is so understudied in climate change, it's ridiculous. In all the climate change literature, there's 1 or 2 or 3 studies, even the authors aren't present. So we're really needing to focus on these areas. North Africa's sweltering, it's boiling to death and it's dry. So we need a lot more work, focus on these areas to see what people are doing. And irrigating the desert might not be the solution.
Our next speaker is Dr. Motswapo Phoko from the Agricultural Research Council. Speaker in person or online? Yes, hi, sorry. It's your right.
Good morning, everyone. My name is Motswapo Phoko. I work for Agricultural Research Council. And this morning, I'll be sharing with you some of the research work that we have done in collaboration with the University of South Africa and the University of Limpopo. And then the title of my presentation is, we were actually looking at the response in terms of the growth performance, methane emission and production, bacterial profiles, as well as the ruminal fermentation of Nguni steers fed Seriphium leaf meal. This is the presentation outline. In South Africa, in our country, the livestock production depends solely on the natural rangeland to meet their daily requirements. But then, however, due to other various number of limiting factors, inadequate supply of good feed being one, they at times unable to meet their production performance, particularly during the dry season when the state of rangeland has deteriorated in terms of quality and quantity. Also, the deterioration of rangeland, the state of rangeland in our country, it's caused by a number of factors, bush encroachment being one, which is also caused by a number of factors. And then for the purpose of this work and presentation, I'll be focusing on one encroacher species that contributes significantly to the bush encroachment rate. This specific plant species has occupied the larger portion of the grassland biomes in South Africa, obviously making the grass production or the herbaceous layer decline in the areas. And then over and above that, the consumption of poorly digested feed by ruminants or by cattle result in cattle losing their body weight condition, their life weight gain, and also emitting the higher enteric methane emission, which contributes to the climate change. So it has been recently the bush encroachment or the woody plant species have gained attention in terms of using them as supplementary feed to improve the livestock productivity. And for this work, we were also looking at, because Seriphium plumosum, also known as Bengra bush, is one of the encroacher species that has taken over the grassland biome in our country. So we were also looking at a way in which we can control the extent of this particular plant species, because there has been many various mechanical and chemical methods that have been put in place to control this specific plant species. And then this is how the species looks. In summer, it's green, wet season. And then in winter, it turns brown, goldish, like when it's dry. And then the map on the far right depicts how infested, how encroached this species is across our country. And then, like I have mentioned, that in this study, we focused mainly on Seriphium plumosum, also known as Bengra bush. So we identified one farm in Gauteng province. This farm is about 550 hectares. Of the 550 hectares, 280 of it is encroached with this plant, meaning there's no grass. So we engaged with the owner of the farm. He gave us permission to harvest this plant. We harvested by slashing the above the ground material, left it on the ground to sun dry for 5 days. Thereafter, we then collected it to ARC in Irene, the experimental site where the experiment was conducted. The far left picture there shows how the plant looked upon being milled, because we collected it and then we milled it at ARC. And the bottle there was when I pulled out a sample from this plant for chemical composition analysis. And there we were formulating the diet using other different ingredients, which I will show you, together with the Seriphium or Bengra bush. This is the experimental diet. We had 4 experimental diets. Treatment 1 did not have any Seriphium at all. Treatment 2 had Seriphium inclusion at 10%. Treatment 3 had Seriphium plumosum at 20%. And the last treatment did not have lucerne hay, which was included in treatment 1 at 30%. But in this treatment, Bengra bush was included at 30%. which means then basically we replaced lucerne hay with Bengra bush. And those were the list of all other ingredients that we used to formulate the diet. Unfortunately, due to time, I did not include the chemical composition of the entire diet, the final diet, because from this formulation, we went back to the laboratory to do some analytical analysis for the chemical composition of each diet. No, sorry. And then this were the diets, the control one, as you can see, you can even spot the maize and the lucerne. Treatment 2, treatment 3, treatment 4, and the above picture there is a plain Bengra bush. And then the animals that we used were sourced from one of our farms in Loskop, one of the ARC, Agricultural Research Council animal production farms in Loskop, is in Limpopo. They were tracked and transported to ARC Irene at the feedlot facility. This work was done at the feedlots, and then this was an intensive study because no study has ever been done using Bengra bush as feed. So this was more like a test study. We wanted to see how the animals will respond upon consuming Bengra bush. So on arrival, the animals from Loskop to ARC, we used 28 Nguni steers, and the choice of the breed was not a factor in this study. We just wanted the animals that are of uniform in terms of breed type, sex, age, and weight, so that when we do the comparison, we are not biased. So upon arrival at the feedlot facility, the animals were offloaded from the trucks and they were allocated into the group pens, 4 group pens according to the 4 treatments diet. So each pen had 7 animals, making them 28 animals, meaning in each treatment was replicated 7 times. We had 4 treatments replicated 7 times. And that was the adaptation period, which lasted for 21 days. After 21 days, the animals were moved from the group pens to the single pens. This was a complete randomized design because the animals were more or less 300 kilogram weight, 22 months old, 28 Nguni breed male steers. So we moved them to the single pen. Each animal was allocated into its own pen, which they had access to water at libitum and also the feed. And then part of measuring the growth performance was to determine the daily feed intake, the daily dry matter intake. We did so by giving in the animals a certain amount. In the beginning, we started off with giving them 5 kilograms, but then as you know, as they grow, they tend to increase the intake. So we would give them a certain portion of intake, and then in the morning when we come in, we take out the leftovers and weigh them. That way we were able to know how much they consumed daily. This study was done for 111 days, inclusive of the 21 days adaptation period, which the initial data was collected for 90 days. And then also to measure the growth rate, we also weighed the animals fortnightly. Every 2 weeks, we would release them from the single pens to the crush pen, where we take we took the weight, and then we would determine the weight gain by subtracting the initial weight from the weight at the time the measurements were taken. We also did at the bottom there, we also measured the body condition scoring. We did it manually by palpating, checking the ribs, with the 5 score being the fat, overfed. We don't want an animal that is overfed, and the 1, which is less, doesn't have much fat, which is also something that we don't want. This is when we were weighing. Also, with the issue of now looking at how Bengra bush can now reduce the methane enteric fermentation, we also measured methane emission using the laser methane detector. This was done for 10 consecutive days in the morning after feeding. For that 90 days, when we were collecting the actual data, we were consistent in terms of feeding. We knew that between 8 and 8:30, all the animals must have received the new feed. So the measurement of methane was taken 2 hours after feeding, which is assumed that at that time, the animals are ruminating. Fermentation is taking place, which is when the animals are then starting to release gas, methane in this case. So this was done by pointing the laser methane detector at the auxiliary part of the animals, that is the nose and the mouth, because it has been said that 1 minute, I'm far, okay. Okay, we also did the bacterial population examination using the DNA extraction. Also, we did the in vitro fermentation because at the time when we were collecting rumen fluid, we were unable to take it straight to the lab. So we had to take this route to replicate it, to do it again. Did this study at the University of Pretoria to measure the rumen fermentation parameters. And then all data was analyzed using ANOVA. This were the results, no significant significant difference was observed across the diet. But as you can see numerically, treatment 2 that received 10% of the Bengra bush numerically performed higher. Those statistically, they were all the same. And also, the DNA extraction 16R genes was able to give us the specific bacteria. We trimmed down from 31 phyla that were detected to at least 10. So because we wanted to see the most dominating bacteria and bacteria that are responsible for methane production. As you can see, the yellow one, Bacteroideota. I'm not sure about the pronunciation. This is one of the bacteria that is associated with lower methane production, which could then explain the reason why there wasn't a significant difference across the diet. And then these were the ruminal fermentation parameters. We measured the total production gas, the in vitro organic matter digestibility, ammonia. We also looked at the volatile fatty acid because they are mostly associated with methane enteric production. And then we then concluded that because there wasn't any adverse effect from the animal fat, Bengra bush, and lucerne, this perhaps could be one of the plant species that going forward, we use it as feed under different production system. Currently, we are in a process of using this plant again at an extensive production using goats just to see how they will respond. And again, because we are battling the issue of controlling bush encroachment in our country, this can in turn help us because the more we feed it to the animals, the lesser we will have it. We want it, I want it, the farmers don't want it. There's always a contradiction between me, myself and them. So this could also be one of the ways, the manner in which we control Bengra bush in our country, because this is one of the species that contributes significantly to the extinction rate of Bengra bush in South Africa. I thank you.
Unfortunately, we don't have time for questions, but I'd urge you to find Dr. Phoko at tea for continued discussion. The next, yes, yes.
For the speakers, please not to stand too close to the mic because there's a lot of popping that comes through through the speakers.
Sure.
Thank you.
Our next speaker is Dr. Reneilwe Harrison from Grasslands Forest Wetland Node, say on Pietermaritzburg. I think your study was from them. Your affiliation now, I'm not sure. Well, thanks.
Thanks, Robin. Good morning, everyone. My name is Reneilwe Harrison, and I'm going to speak to you today about a very short part of the study that we conducted on the environmental factors influencing dissolved organic carbon concentrations in Afro-montane grassland catchments. So our study aimed to gain an understanding of the various environmental factors which drive the temporal patterns of DOC exports in Afro-montane grasslands. The reason why we wanted to do the study is because literature has attributed DOC to climate, hydrology, land cover and management, different soil types, as well as topography. So our study was based in the Cathedral Peak research catchments. These are managed by SAON. And yeah, Cathedral Peak is part of the Drakensberg mountain range, which I think we all know is between on the border of KwaZulu-Natal and Lesotho. We chose two different catchments to do our study. One is called Catchment 6 and the other is called Catchment 9. Catchment 6 is managed as a grassland system. It's burnt every second year. And Catchment 9 has been excluded from fire mostly and is actually quite a woody catchment. So what we did was we installed piezometers throughout the catchment areas. I'll just go back. So we tried to install piezometers in the wetland systems. So we had them up at the top of the catchment, in the middle of the catchment where the majority of wetlands are in Catchment 6, and then again at the base of the catchment near the weir. And then again in Catchment 9, we tried to find the wetland systems and install the piezometers there. So yeah, we had piezometers throughout the catchment areas, and then we also had two high frequency probes which measure DOC every 15 minutes. And we installed those in the weirs of both the catchments. And then we sampled water from both the piezometers and from the high frequency probes over a period of about 2.5 to 3 years between 2019 and 2022. And then we compared the results of the DOC against daily rainfall, water temperature, turbidity, conductivity, and stream flow discharge. And what we found, just one of the things that we found was that DOC concentrations were a lot higher in the wetland areas versus the terrestrial areas in the soils of those areas. And then we also found that DOC follows seasonal trends of precipitation. And what we also found was that DOC concentrations are dependent on varying hydrological conditions and how these relate to the flow paths of a catchment area. So the DOC follows the connectivity of flow paths between wetlands and stream networks. Yeah. So this was just one of our findings. When we started the study, it was in 2019, towards the end of a drought period. And what we have is, so this is Catchment 6 and Catchment 9. What we have is very low rainfall in the beginning of 2019, corresponding very low stream flow, and very low DOC flux rates coming out in the stream flow. In about February 2020, we got a nice amount of rainfall, which sort of broke the drought period. And then we had a corresponding increase in stream flow and then a massive spike in DOC flux rates coming from Catchment 6 and then again from Catchment 9. And then the DOC flux rates and the stream flow obviously decreased as winter came along in 2020, and then they increased again as the rainfall began in spring and summer. And then, yeah, so it followed the seasonal trends of the rainfall. But interestingly, what we found was that there was a lag time effect between when we got a stream flow, when we got an increase in rainfall to when we got an increase in the stream flow. There was a period of about a few days to a few weeks, depending. So we got an increase in the rainfall, and then a few days or a few weeks later, we got an increase in stream flow and a corresponding increase in the DOC flux rates. And what we found was that as a result, from the piezometers, as well as from mapping the different soil types in the catchments, we found that what has to happen is that the wetlands have to fill up with water. They have to become saturated again, and they are attenuating all the DOC. And once they become saturated, they start producing overland flow. And then that overland flow carries the DOC into the stream. So the amount of DOC flux rate that's coming from the wetlands, amount of DOC flux rates in the streams comes from the wetland systems. And we have to wait for the overland flow to start being produced from the wetlands to carry the DOC into the stream. So just in conclusion, we found that DOC concentration variability is closely linked to the drying and wetting cycles of wetland systems. These influence both the base flow connectivity as well as the overland flow during rainfall periods. So the connectivity of wetlands to streams within both catchments plays a vital role in the attenuation and export of DOC within these watersheds. And I think from the study that we need to think more about biogeochemical cycles in land use management, and it's often not thought about. But yeah, I think it's definitely important. Thank you so much.
It's a question for Heidi.
Hi, Heidi.
Oh, thanks, Reno. I think this is such important work at this landscape scale. I just wondered how costly those piezometers are.
The piezometers themselves weren't costly at all, but the high frequency probes were very costly.
Oh, okay.
The piezometers it's just a PVC pipe.
Yeah.
And we installed them into the wetlands.
And then the probe
and
measures.
the probes are very expensive. Sue knows. Sorry.
Okay.
Yeah, we got them from University of Burgundy in France. So I don't know how much do they cost you. Yeah. Okay. Yeah. Thank you.
Thanks, everyone.